Hello, my name is Molly, and I'm going to be talking about 3D cell cultures and the information I found about them. So, in class, we have talked about 2D cell cultures where the cells are grown in a flask that lays flat, and how they allow researchers to grow cells and tissues outside of an animal or human. However, 2D cell cultures have a limitation because they do not create the exact environment as in vivo. And 3D cell cultures are an expansion of this because it involves creating a 3D extracellular matrix to allow cells to grow in every direction and with the same conditions as natural in vivo environments. And this technique has been used in cancer research. There are three types of 3D cell culture methods. One is scaffold-based, where the cells are supported by hydrogel-based scaffolds, and the hydrogels can be either natural, such as collagen, or they can be made by synthetic polymers. The second type is scaffold-free, where the purpose is to make the cells aggregate into spheroids. And one type is hanging drop microplates, which are plates that have open bottomless wells. And this allows the cells to grow through the bottom and create spheroids. And the second type is low attachment plates, which are coated with hydrophobic or hydrophilic polymers that encourage the cells to grow away from the wells and aggregate with each other. And then there are also specialized 3D cell culture models, which include micropattern or microfluidic devices. In this figure, the researchers evaluated mesoporous silica nanoparticles in a nanocomposite hydrogel. C is a confocal microscopy image of the matrix they created with collagen, and it shows the distribution of the MSN cells, which are read inside of the matrix. This figure shows scanning electron microscope images of pancreatic cancer cells. Through CD cell culturing and formalin, they formed spheres and created highly detailed images. This figure is from a paper where the researchers wanted to make a 3D endometrial tissue model. So they took epithelial and stromal cells from rats and made layered cell sheets and placed them in a collagen gel. A is an actual picture of the layered sheets, B is the layered sheets after staining, and C on the right is an immunohistochemical stain. The advantages of using 3D cell cultures instead of 2D cell cultures is that one, it creates a more accurate representation of the natural environment of cells, and it allows researchers to see not only cell-to-cell interactions, but cell-to-matrix interactions. It shows cellular functions, and cells have access to more nutrients and oxygen. The disadvantages is one is obviously more expensive to use. Two, it takes longer for the cells to fully grow. And three, forming the 3D matrices takes more time and resources.